Research and Development Laboratory. Swaroop's product range includes bioorganic fertilizers, natural and certified organic products, micronutrients, 100% natural herbal pesticides, special inorganic agrochemicals, and pesticides. Swaroop manufactures around 50 products which find application in almost all types of field crops, vegetables, cash crops, floriculture, and horticulture crops. Now, let's have a look at this audio visual.
हे आहे माझं होम गार्डन माय ग्रीन थंब आणि माझी सगळ्यात फेवरेट प्लेस
आणि ही प्लेस फेवरेट बनते स्वरूपच्या होम गार्डन शॉपिंगमुळे.
आपल्या मुलाबाळांप्रमाणे झाडांची काळजी घेणारी
20 वर्षांपासूनची शेतकऱ्यांच्या विश्वासाची स्वरूप होम गार्डन शॉपिंग.
कम्प्लीट प्लांट केअर.
Before starting with today's event, let's have a quick look at what is National Pharmacy Week. National Pharmacy Week acknowledges the efforts of pharmacists for improvising healthcare facilities and creating awareness about the role of pharmacists in the society. IPA organizes National Pharmacy Week during the third week of November every year. It was created to recognize the vital contributions that pharmacists, student pharmacists, and pharmacy technicians make every day to patient care. It emphasizes on creating awareness about the national pharmacy theme in specific and about the pharmacy profession and the role of pharmacists in general. The theme of National Pharmacy Week this year is Pharmacists, Frontline Healthcare Professionals, and is being celebrated throughout the country from 16th November to 22nd November 2020. This theme is highlighting the role of pharmacists amidst COVID-19 global pandemic. Community pharmacists and hospital pharmacists, along with their teams, are vital healthcare providers during this outbreak, as they remain on the front line of public health while serving as direct points of access for their patients. They play a crucial role in their infection control. This is the third year we are celebrating National Pharmacy Week, and we have organized pharmacy communal sessions for 3 days, elaborating various topics related to the field of pharmacy. The topic for today is vaccine development process, which will cover further subtopics like need, discovery, manufacturing, and clinical testing of vaccines.
It is a great honor to be a part of this medical sector, which is filled with talented and dedicated healthcare professionals who are indulged in improving the patient care system. Today, we have with us an eminent speaker with a 15 years of teaching and industry research experience, Dr. Harshal Pawar. He is currently working as an assistant professor at Dr. L.H. Hiranandani College of Pharmacy. He has also worked as a head of department of quality assurance for 6 years and has worked with various pharmaceutical industries like Medley, Charak, and Alchem Research Center. He has published more than 60 research and review articles and has also presented various research papers in national and international conferences and seminars. To his credit, he has 2 Indian, 1 American, and 1 international patent. Not only this, but also he has received grants from Mumbai University and is on the editorial board and reviewer of national and international journals. Nevertheless, he has also guided 25 MPharm research scholars in the subject of quality assurance. He is also recognized guide of Mumbai University and Pacific University. Along with this, he has also authored 8 books in the field of pharmaceutical sciences and is a life member of Association of Pharmaceutical Teachers of India. Sir, we are highly obliged to have you here today and on this platform. Now, I request the Vice President of IPA-MSB and our mentor, Dr. Anand Shedge, to felicitate our speaker of the day, Dr. Harshal Pawar. Sir, you may begin with the session. You have been made the presenter.
Hi. Let me share my presentation first. Can you allow me to share?
Yes, sir.
This is
Yes, sir.
Sir, I guess you have been given the permission to share your screen.
just a minute.
Yes, sir. Yeah. And sir, your screen is visible.
But I'm unable to share my presentation. I'm not getting that option actually. Can you just
Actually,
guide me?
Sir, we can
yeah.
view your
Actually,
screen.
we can see your screen. So I guess you just have to open your presentation now.
Okay.
Yeah.
Can
Yeah,
you
it
able
is
to
visible.
see my presentation?
Yes, sir.
Okay.
It
Yes,
is
Okay.
sir.
Fine. Okay. Thank you very much, Shruti, for giving such a nice introduction. At the same time, I would like to congratulate Indian Pharmaceutical Association for celebrating this 59th National Pharmacy Week. At the outset, I would like to say thanks to the President, Dr. N. Shiva Prasad, Vice President, Dr. Anand Shedge, then Student Exchange Officer, Mr. Gaurav Zope, and Student Coordinator, Rahul Jaiswal, for giving me this opportunity to interact with all of you. The topic of my today's talk is vaccine development process, and I think I have been given 90 minutes time, so I'll try to wind up the whole presentation within those 90 minutes. These are the contents of my today's presentation. Okay. So these are the contents. First, we'll start with the general introduction about the vaccine. Then we're going to discuss the challenges in the development of vaccine, followed by steps involved in the development of different types of vaccines. Then we're going to discuss the manufacturing and quality control aspect of the vaccine. This will be followed by the regulatory requirements and the current regulations which are applicable, especially for production and manufacturing of the vaccine. And I know that everybody is interested in knowing the status of COVID-19 vaccine. So a little bit I'm going to talk on that COVID-19 vaccine. So let us start the presentation. I hope you must be knowing this particular terminology, biologics or biological products, right? So you will see the official status. So in most of the pharmacopoeia or you will see most of the reference standard reference books, right? So you'll come to know this particular terminology, right? So vaccines basically they are covered under this particular broad head, that is what biologics or biological products. Let us see what is vaccine. So a vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. So typically these vaccines, they are made either from a live germ or microorganisms or killed form of microbes, or sometimes some of the important subunits or components, or you can say toxins, which are secreted by certain bacteria or viruses, or even some of the components like surface proteins. Let us see how do these vaccines they work. So after vaccination, the antibodies they are going to secrete in our body, and these antibodies they will going to fight with the infectious disease, or you can say the pathogen, right, which is coming in contact with our body. Basically, two important type of active acquired, or you can say adaptive immunity that is involved, especially here. That is the first one is humoral immunity, right? So whereby the antibodies they are secreted from the beta cells, and second one is cell mediated immunity. So wherein the T cells they are playing a key role in initiating or in stimulating the immune system, right? So sometimes they are working alone, or sometimes both these immune systems they are involved in fighting the disease. These are some of the licensed vaccine which are used to prevent various diseases which are caused by several viruses and you can say bacteria, right? So some of them are you can call them as monovalent vaccines. Some of them are polyvalent vaccines. And some vaccines they are even available in the combination forms. Let us understand first what is the difference between vaccine and medicine. Right? I hope you must be knowing this difference. That medicine basically they are used for treating specific disease or a disorder. Whereas when talking about vaccines, so vaccines in initial days they were used to prevent a particular disease, or they were used for offering the protection or for prophylaxis purpose. But now if you look at the current trend, the vaccines they are also used for the therapeutic purpose. So this is what the main difference between vaccine and medicine. Let us see the similarity between vaccine and medicine. So vaccines are also a medicine. They do have potential for adverse effects or side effects. They are also made up of multiple ingredients or components. They do have potential for interacting with various diseases or medicines, and most importantly, they need to comply with the standards for safety, efficacy, and quality. So all those standards which are applicable, especially for medicines, I will say even more stringent norms, regulations, and requirements they are applicable, especially in the development of vaccines. So these are the normal routes of administrations which are generally used for some of the previously licensed vaccines. So the first route is oral route. Then some of the vaccines they are administered even by intramuscular route, then subcutaneous route, and even intradermal route. Even the route of administration is not just restricted to the routes what I have mentioned, but many research and investigations currently they are going on for administering the vaccine from the different routes. Right, here I have presented a few examples. You can see there is one photograph of influenza virus vaccine. So this is inactivated type of vaccine, and this particular vaccine is administered through the patches. Right. So one more drug delivery is there. That is what intranasal drug delivery. Right. So the same influenza vaccine that which is available in the form of live viruses or a live attenuated vaccine. So that is administered through this intranasal route. while developing the vaccine, the development scientist has to keep the proper balance between the efficacy and safety. If you look at the previously published literatures and reports, so you'll come to know that many adverse events or adverse reactions or side effects have been encountered by the previously developed licensed vaccines. Since vaccines, they are not studied for their long-term effects, right? So there may be chances that there may be some unforeseen adverse events they may came across after a certain period of time. And there is no accumulating testing, or you can say no testing regarding the carcinogenic, mutagenic, or the potential reproductive effects of vaccines. So all these data, particularly it is collected during the phase 4 trial, that is what post-marketing surveillance. So based on the severity, the adverse reactions, they have been classified into 5 different categories. That is class 1, class 2, class 3, class 4, and class 5. So if you look at the type of reaction which are given out under class 1, so those are especially not related to the vaccine, but the procedures which the health worker generally followed during the immunization or vaccination. Right, so sometimes the dilution may not be proper, sometimes the syringe is contaminated, vaccines are not stored proper, so they also gives rise to a certain kind of adverse events or adverse reaction. The second class, these are local reactions like lump or swelling at the site of injection. The under the third class, facial swelling is there. Some more severe adverse reaction you can find under class 4. That is what systemic signs, then fever, vomiting, diarrhea. If you look at class 5 adverse events, then that includes a severe hypersensitivity reaction. That is what we are calling it as anaphylaxis. Right. So sometimes it may lead to a shock, collapse, and death. Right. So this indicates that the development scientist or formulator of the vaccine has to take care of all these minute things while developing the vaccine. Let us look at the important milestones in the vaccine development. If we look at the 19th and 20th century milestones, then initially the Peptide vaccine, it has been introduced, followed by the first combination vaccine that was introduced in 1948. If you look at the era of around 1915, then the polio vaccine, the oral polio vaccine was introduced. In 1917, the first polysaccharide vaccine, followed by a conjugated vaccine that was introduced in 1980. And in 1981, the first recombinant antigen vaccine was introduced. Just have a look at the 21st century milestones. So the first important contribution, it was done in 2008, wherein the first mRNA vaccine, it was developed. This is followed immediately by the introduction of first therapeutic vaccine, which was developed to treat the prostate cancer. One important technology, it was introduced in 2013, that is what reverse vaccinology. It has given a new direction in the development of this vaccines. And in 2013-14, the peptide vaccine, this particular category, it was introduced for both prophylaxis category as well as therapeutic category. These are some of the important challenges which are involved in the vaccine development process. The first and most important challenge is in understanding the mechanism of developing the immunity, right? So you must be knowing that the immune system, it is a very complex system. Most of the organs, tissues, cells, a very complex system is there, so it is very difficult sometimes to understand the appropriate immune response exhibited by a particular vaccine. So during testing, basically here the major challenge which is first. The second challenge is due to the host variability, right? Even in a current COVID situation, you must have read out somewhere in newspaper, even in some of the literatures, that a lot of host variability. So the difference in the response to the coronavirus has been observed with the people from the different location, even with age, with sex. Right. So in case of coronavirus, also we have seen that the older people they are more susceptible or prone to the infection. Right. So even to know the inter-individual variability, that becomes sometimes difficult because of the variation in the genetic composition of everyone. Next important challenge is the pathogen variability. So we have different kind of pathogen of having a complex biology. Even you can see that two different strains of the same species, right? So if you look at their genetic sequence, so you can get a lot of difference. The next challenge is the vaccine safety. So as I said earlier, that it's quite difficult, especially in the pandemic situation, to determine the what you can say the projected side effects or unforeseen, unwanted adverse events at a very early stage during the vaccine development process. So apart from this, the environmental and geographic factors like poor nutrition, obesity, and even co-infections, then prior immunity, pollution. So all these factors, they are also having a great impact, especially in the vaccine development process. These are some of the important ideal qualities which are expected from the vaccine. So the first one is the vaccine should have ability to stimulate the appropriate immune response for the particular pathogen. It should be effective in preventing or reducing the severity of infectious disease. It should achieve immunity with minimal number of doses. It should provide long-term protection. So this is what the prime requirement. Then it should provide the maximum number of antigen that confer the broadest protection against infection. Right? So that is the reason that in market we are getting polyvalent vaccines or multivalent vaccines, right? So sometimes the more number of serotypes they have been incorporated, or antigens have been incorporated, so that it will help to get the strong immune response. Even that whatever vaccine is developed, that should be stable, especially during handling and storage. Till the time of, I'll say, immunization or vaccination. And one more thing is that the vaccine should be inexpensive, so that should be cost effective, right? So it should be cost effective, especially to the common public. And it should be available for general use through the mass production. And last but not least, that is, it should be safe in terms of side effects or adverse events. So it should bear a minimum side effects or minimal, you can say, adverse events. Right? So let us have a look at the different types of vaccine. So majorly the vaccines, they are categorized into four major categories. The first one is live attenuated vaccine, second is inactivated vaccine, third one is subunit vaccine, and fourth one is DNA and RNA based vaccines. So let us discuss one by one. The first one is live attenuated vaccine. So the term itself indicates what these vaccines are, what it contains. So it contains live microorganism. Attenuated means weak. So weak in terms of its virulent property, disease causing property. Right. So these vaccines basically they are produced from the pathogen or from the microorganism which is causing disease. Right. So its virulence property or disease causing property, it is being reduced, or so you have to make that particular microorganism non-virulent. Right. So but at the same time, one has to retain the replication property, multiplication property of that microorganism, right? And that is the reason in case of live attenuated viruses, vaccines, being the live microorganisms are there and they have an ability to multiply in the body. So we are getting long-term protection from this type of vaccines. The second type is inactivated type of vaccine. So this is again one more conventional type of vaccine as like what live attenuated vaccine. So here you need to inactivate, or in a simpler term, you need to kill the germ or microorganism. And these killed germs or microorganisms, we're going to use it in the vaccine. But in this case, being the microorganisms or germs, they are inactivated. At the same time, their virulent property, it is destroyed. Being these are killed microorganisms, they won't multiply in your body, and therefore the booster dosage or more number of shots of the vaccines, they are required in order to get the protection or immunity for longer duration of time. One more important thing is that to enhance the immunogenicity or immunity of these particular vaccine for longer duration of time, one can even add some more agents like adjuvants. Let us have a look at the third important type of vaccine, that is what subunit type of vaccine. So the term here itself indicates what is subunit. So it is a piece of microorganism or germ which cause the strongest response, right? So this can be any component, any part of the microorganism, provided that that particular part is having ability to function like antigen, right? So in response to that antigens, the antibodies, they are produced, and then you will get the protection. Here also many a times the adjuvants, they are combined along with the components of the microorganism, right? So here we can take the example of coronavirus. So on the surface of coronavirus, you can find the spike proteins, and these spike proteins, even you can see in the picture, right? So the spike, these proteins you can purified, isolated, purified, and you can put it into the vaccine, right? So that is what subunit type of vaccine. And next one is a DNA or RNA type of vaccine. So here, I hope you must be knowing that DNA or RNA vaccine. Basically, these are the specific kind of proteins or the genes. We can say these are the specific gene or genetic material, and they will going to produce the required components, or I'll say required proteins, which will behave like an antigen, and in response to those antigens, our body will going to produce the antibodies, and then will going to get the protection. Right. So here also the adjuvants, they are added to enhance the effectivity or immunogenicity of the vaccine. So among all these four different types of vaccine, currently the mRNA vaccine that is found to be more safer and cost effective, and even it is very easy to prepare as compared to other type of vaccine. There is one more kind of vaccine that is called as toxoids. I hope you must have heard about these toxoids. So these toxoids, they are prepared from the toxins which are secreted by the microorganisms. As of now today, most of the toxoids, they are available in the market for the bacterial toxins. So if you'll look at viruses, so hardly I think till date only one vaccine, that is what rotavirus, have been identified, which is secreting the toxin. So mostly the toxoids, they have been developed for the bacteria or bacterial infections. These are some of the examples of the previously licensed vaccines. So for live attenuated vaccine, tuberculosis, that is BCG vaccine is there. OPV, that is oral polio vaccine, measles, rotavirus, yellow fever. For inactivated or killed antigens, whole cell pertussis and inactivated polio virus for subunit type of vaccine. So sometimes they are also called as purified antigens. These includes your pertussis vaccine is there, influenza type B vaccine, hepatitis B vaccine is there, and the toxoids, tetanus toxoid and diphtheria toxoid. Now let us see what are the different approaches or ways which are available for the development of vaccine. So as I discussed some of the types of vaccines, so whole inactivated virus or microorganism you can use, than a live attenuated virus or microbes you can use. Here sometimes virus like particles you can use, right? So ideally they are not a virus, or they are not a microorganism, right? But they are looking like that. One more approach is there, that is what recombinant technology, right? So I think nowadays this recombinant technology, so many vaccines in the market, they are coming out with this particular technology. So here sometimes the vectors, they are used to carry a particular gene, right? And apart from this, the individual you can say DNA vaccine is available. Then you can even go for the synthetic peptide. So this is one more approach, right, wherein just by knowing the sequence, you can able to synthesize a particular component or particular part of the protein, that is what peptide, right? And this peptide you can fill it into the vaccine, and these peptides will going to induce the immunity. So let us discuss this approach one by one. So first I'm going to discuss the conventional approach, wherein first we'll start with the development of live attenuated viral vaccines. So the development of this particular vaccine, it is based on one fundamental, that virus specific for a human may become attenuated by passage in non-human cell lines, right? So the virus or the bacteria or the microbe which is infected human, right? So you'll have to pass it through the several cell lines of non-human, or you can say animals, right? And during this phase, what will happen? The pathogen or the virus who is causing a disease to the human being, right? So that will undergo mutation, and after a certain period of time, that particular pathogen it will become non-virulent, especially to the human cells or human being, right? So then it becomes a good candidate for developing the this live attenuated type of vaccine. But the disadvantage of this particular process is that so it takes a lot of time, even several years it takes to develop. Right. So this is what the process here I have indicated, which is used for developing the live attenuated vaccine. So while viruses, bacteria attenuated by repeated passing the cell culture or animal embryos. Right. So during this period, here the period which is specified, that was 10, even more than 10 years. Right. So during this phase, you'll have to pass the particular germ through the several cell lines, right? So we'll have to do the subculturing several times, right? So during this particular period, the mutation is taking place, the germ is becoming weak in terms of its virulent ability, right? Then you'll have to prepare the vaccine by using this non-virulent strain, and then you can do the vaccination by using those vaccines. So those non-virulent bacteria or viruses, they will going to replicate in the host body, and there will be an induction of immune response, and the individual or the host will get the protection against that particular disease. So here I have quoted some of the examples. You can look at the live attenuated chickenpox vaccine, then yellow fever vaccine, and influenza vaccine. Just go through their developmental stages quickly. So you can see here, in case of chickenpox vaccine, so around the clinical isolate of the pathogen has been isolated first, and it is passed through around 11 passages through the human embryonic lung fibroblast cells at around 34 degrees Celsius, followed by 12 passages through the pig embryo fibroblast at around 37 degrees Celsius. Then 5 passages in human embryonic lung cell lines. And then finally, you are getting that non-virulent strain, right? And then you can use it for preparation of vaccine. So similarly, here also you can see that how many number of passes have been taken to develop that non-virulent strain. There is one more way to prepare the live attenuated viral vaccines, that is what recombinant DNA technology, right? So here you are going to take the you will going to isolate the pathogenic virus, and then you will going to determine its genetic sequence, or you can say the sequence of the entire genome. The virulent gene, it is being identified, right? And this virulent gene, right, it is deleted, right? So sometimes you can delete this virulence gene, right? So keep in mind that the other components of the virus that will remain as it is unaffected, especially the epitopes, those should not get affected here. In case of there is one more way, that is what you can subject this particular gene, that is for a mutation, that is what mutate the virulent gene, and through this also you are getting the non-virulent viruses, and these you can use to prepare the vaccine. Of course, there are certain positives and negatives for live attenuated vaccine. So the duration or time what is required for development of this live attenuated vaccine, it is more as compared to the other categories. You just look at the positives. So it gives excellent immune responses. The viruses or bacteria, they will remain live, so they will undergo multiplication. We will look at the negatives in terms of the safety and stability. These vaccines, they are less safe as compared to the inactivated vaccine. The reason is that the attenuated pathogen can revert to original form and may cause disease, right? So this is the major drawback of this vaccine. Then one more drawback is that during processing of these vaccines, so there may be chances of contamination in the tissue culture, and of course some more drawbacks are there. Right. So if we'll see at the current development, especially live attenuated vaccines for COVID-19. So currently the scientists, they are not focusing on live attenuated vaccine that contains particularly the SARS coronavirus 2 virus. But instead they are testing the previously generated live attenuated viruses for common cold and measles viruses. But simply what they are doing, they are just manipulating the genetic code of these previously generated attenuated viruses, so that these viruses will going to display the key SARS coronavirus 2 spike protein. And so you can classify this particular category of vaccine under the subtype of this live attenuated vaccine, that is called as viral vector vaccine. So such three vaccines, they are in pipeline. One of them is measles vector vaccine, that is under preclinical trial, and it is undertaken by University of Petersburg. One more is there, that is HD 1 to 22. That is under development. It is under phase 3 trial. That is what you can say. That is developed by University of Oxford. One more is there. That is what adenovirus 5 and coronavirus. So not coronavirus. That is what common cold virus, and it is in phase 2, and this is being developed by CanSino. This is what the second type of vaccine, that is inactivated whole cell vaccine. So as I said, here you will going to inactivate the virus, or you will going to kill the virus. So different techniques you can use to kill or inactivate the virus. So you need to subject the virus or the germ or the bacteria to different stress conditions, right? So that includes, you can even change the pH, you can bring about dehydration. Sometimes you may do agitation, even sometimes metal ions, they have been used to inactivate the germs. Even you can change the temperature, so freezing or even heating you can use for inactivation. The best technique, whichever is used, that is the radiation technology. So here the low energetic electron irradiation or radiation, they are used to inactivate or to kill the vaccine. You just look at the picture here. So you can see here the intact virus, right? And at the surface of this intact virus, the antigens are there. So after killing or after inactivating this particular virus through low energetic electronic irradiation, the antigen portion, the surface protein, the spike protein in terms of this coronavirus, right? So they will remain intact, whereas the genetic material like nucleic acid, it is destroyed during this process, right? So one has to take utmost care while inactivating the particular germ, that the epitopes, the component, the protein which is acting as an antigen, which is responsible for induction of the immunity, that should remain intact. One more thing, you can see here the nucleic acid or the genetic material, it is destroyed, so the cells will not able to multiply, and that is the reason that in case of inactivated or killed vaccine, the booster doses, they are required for getting the long-term response. In terms of safety and stability, these vaccines, they are more safe and more stable as compared to the live attenuated vaccine. So here I have mentioned two examples of COVID-19 vaccines from this particular category which are under development. One is inactivated vaccine that is under phase 3 trial, which is developed by Wuhan Institute of Biological Product. One more is there, that is CoronaVac, which is a phase 3 trial that is by Sinovac. The next category is a subunit vaccine. As I said earlier, it is also called as purified antigen. So here, basically, the proteins or sugar component which are present on the surface of the vaccine, mostly they are isolated and they are used as an antigen. Here, the identification of that particular subunit or that piece of the microorganism or germ, it is very, very important. If correct piece is selected, then I think you can able to develop a highly effective vaccine. So there are three different kind of subunit vaccines. So first one is protein-based vaccine. So here you are going to use the surface proteins for developing this kind of vaccine. Polysaccharide type of vaccine. So this kind of vaccine, especially it is developed for the bacteria, because you must be knowing that the bacterial cell wall, mostly it is in case of capsulated bacteria, so it is coated with the polysaccharide. So it is also having immunogenic property. So that is used to prepare the vaccine. And third one is conjugated type of vaccine. If we will compare the positives and negatives, first I'll discuss the positives that since there is no live component of the germ or microorganism, so these vaccines, they are comparatively more safer. Even they are more stable as compared to live attenuated vaccine. Of course, one major drawback is there that these vaccines, they are not as strong as what the strong immune response which is exhibited by the live attenuated vaccine. Mostly the recombinant technology, it is used for developing the subunit type of vaccines or even DNA-based vaccine or vector-based vaccine. So wherein the first the pathogen it is being observed, it is identified, then its genome sequence is determined. The particular gene which is responsible for causing the disease or for acting as an antigen, that is being identified, and then it is cloned. Then the recombinant construct is being prepared, and from there the DNA vaccine, subunit vaccine, and vector-based vaccine, they are prepared. So first we'll talk about the recombinant vector vaccine. So here, so as I said earlier, so you'll have to identify a particular unit or a particular protein, or I'll say a part of the gene which is responsible for immunoprotective property from the virulent virus or the germ, right? So you're going to encode that particular gene, then we'll have to incorporate this protein or this gene into a carrier or a vector which is non-pathogenic. Here that particular gene, it is incorporated, and then by using these non-pathogenic viruses, or I'll say this viral vector, it is used to prepare the vaccine. Right? So once the host or the individual it is vaccinated, so these viral vectors, they will enter into the body, and these viral vector they will stimulate our body to produce certain kind of proteins or certain components or certain antigens, right? And in response to those antigens, the antibodies will get prepared, and one can get the protection. The most important advantage of this recombinant vector vaccine is that the risk of inversion of virulence that is being eliminated through this particular process. And these vaccines, they are providing us both cell-mediated as well as humoral type of immunity. The second way of producing the subunit vaccine, that is what recombinant subunit vaccine. So here you're going to identify and isolate a specific gene from the virulent bacteria or viruses. Then this particular gene, it is inserted into the plasmid DNA and ligated with the ligase. So this new engineered plasmid you need to insert into a bacteria. So this particular process is called as transformation. Right. So these bacteria or the germs you need to grow in a particular environment. Right. So after multiplication or during the multiplication, the antigen, these bacteria or these germs, they are going to produce some proteins, or you can call them as antigens. So these antigens, they are separated and they are further purified, and they are incorporated into the vaccine. So you can see here one example I have put it, that is of recombinant hepatitis B vaccine. So here you can see the hepatitis B virus. So here the particular antigen producing gene, it is being isolated from this virus. And on another side, you can see here the one bacteria is there. From this bacterial cell, the plasmid DNA, it is being isolated. It is cut with the restriction enzyme, right? And it is then combined with this antigen producing gene, which is isolated from the hepatitis B virus. So here you are getting the recombinant DNA. This recombinant DNA, then you need to incorporate it into the yeast cell, right? And this recombinant yeast cell, you need to subject it to the fermentation process. So during the process of fermentation, the recombinant yeast cells, so they will going to so you can see here certain antigens or the proteinaceous substances, they are being secreted by these yeast cells, right? So in the fermentation tank. So these secreted antigens or proteins, they are isolated, extracted, and then they are purified, and after purification they are incorporated into the vaccine, and then these vaccines, they are injected in our body. So this is how one can prepare the subunit vaccine by the recombinant technology. There is one more type, that is what conjugated vaccine. So this is you can call this as a conventional subunit vaccine. So as I said earlier, that the bacterial cell wall, basically, if you look at the capsulated bacteria, so their outer coat, it is mainly made up of polysaccharide, and these polysaccharides, they are having immunogenic property. But initially, simple polysaccharide vaccines have been developed by using this polysaccharide, but being those polysaccharide vaccines, they are having poor immunogenic property, right? So these polysaccharides have been linked with certain proteins, right? So again, these proteins, they are obtained from sometimes as a toxin, or sometimes as a component of a particular germ or microorganism, right? So both these polysaccharide and proteins, they are linked, and finally you are getting conjugated vaccine. So one such a vaccine recently it was developed, that is what typhoid conjugate vaccine, which is more effective and preventive, especially in treating typhoid fever and the children which are under age of 5 years. So you can just look at the manufacturing steps. So here, initially the bacteria, it is subjected to the process of fermentation, where in the polysaccharide, it is being isolated, it is purified. At the same time, in another fermentation vessel, you can see the another bacteria, it is fermented. A particular protein from this bacteria, it is isolated, right, and it is further purified, and after purification, this protein, it is chemically cross-linked with the isolated polysaccharide, right. So finally, you are getting the conjugated polysaccharides, and these conjugated polysaccharides then further purified. So finally, you are getting the conjugated vaccine, right? And these conjugated vaccine, then they are injected into the humans. There is one more type of vaccine, that is what synthetic peptide vaccine. Here the peptides they have been synthesized in vitro, and these peptides, they induce the protective immune response when administered into the host. So these are short chain peptides, and they act as surrogates for the immunogens. So basically, these short peptides, they are representing the fragment of the protein antigen sequence, which are generally present in the positive microorganism. Why to use the synthetic peptide vaccine? So there are several advantages. So in case of synthetic peptide vaccine, they are chemically well defined, they are highly selective, and that too, they are very safe. They are stable at ambient temperature. Normally, for storing the subunit vaccines or live attenuated or even inactivated vaccine, if you'll see the recommended temperature, it is around 2 to 8 degrees Celsius. But in case of peptide vaccine, they are more stable, and even you can say you can store it at even room temperature. So no cold chain requirement is there, and hence they are highly cost effective, especially in the tropical countries. And one more advantage is there, that they are very simple to prepare and very easy to standardize. Right? The reason is that they are chemically well defined, so there is no problem, especially in developing the method of standardization. Right? So this is the methodology which is followed for production of synthetic vaccines. So here you need to first isolate the pathogen, right? You'll have to identify the target protein. On this targeted protein, you need to identify the targeted gene or a particular what you can say the epitopes or the pattern of peptide, which is especially responsible for its pathogenic property, right? So you need to study the genetic sequence, right? So find out the genetic sequence, and by knowing that genetic sequence, right, you need to synthesize that particular component, that particular part of that particular peptide in the laboratory, right? And this synthesized peptide, then you can incorporate it into vaccine. So most of the times, along with these peptide, synthetic peptides, some more adjuvants are added to increase the immunity potential or immunogenicity of these vaccines. So these are some of the important benefits. So the composition is fully defined, as I said earlier. So large scale production, it is affordable. As I said earlier, so storage, so they are stable, especially at room temperature. You can even very easily freeze dry them. Even there is no question of biological contamination. Even these synthetic peptides, they are also having the therapeutic application. So even some of the peptide vaccines or even synthetic peptides, they are used for different therapeutic purposes, especially in developing the vaccines for the cancers. Of course, there are some weaknesses of these peptide-based synthetic vaccines. So they are having poor immunogenicity, and that is the reason, as I told earlier, that adjuvants, they are mixed along with these peptides. No doubt, in vitro, outside the body, these peptides are stable, but in vivo, inside the body, they are less stable. There may be chances of loss of native conformation under some of the stress conditions, and they are effective for a limited population. Of course, some solutions are there to improve these peptide-based synthetic vaccines. We'll see currently these are the vaccines which are under development. Those are falling under the category of subunit type of vaccine. So the first one is by that is what AD26CoV2S. That is what it is under phase 2. I think now it is under phase 3 clinical trial that is prepared by Johnson and Johnson, and 2 more are there under the development. Additionally, here the scientists, they are investigating a specific subunit vaccine that one can use it like virus-like particles, so which will display the particular properties of coronavirus to the immune system. So accordingly, the immune system will react to those particular vaccines. So here the virus-like particles, they are having membrane similar to the actually which is there in case of SARS coronavirus 2. But the genetic code within the particle, that is missing. And that is the reason they are not infectious, right? So the next category is DNA or RNA type of vaccine. So I think you must be knowing that in cell, the genetic component, that is what DNA, it is present, and you must be aware about 2 important processes. One is transcription, and second is translation process. Right. So from DNA through the transcription process, the RNA is synthesized, and from RNA through translation process, the proteins are synthesized. Here, all the 3 candidates, first one is DNA, second one is RNA, and third one is the proteins. So all these 3 components, they are used to induce the immunity. Right. So in case of DNA and RNA vaccines, these DNA and RNA, when they are entering in the host body, so they will direct the synthesis of some specific kind of proteins, right? So these proteins, they are having immunogenic property, and in response to that, the antibodies will going to develop, and you will get the protection. It will compare both DNA and RNA vaccines. So as compared to DNA vaccine, the RNA vaccines, they are more prepared. The reason is that they are very easy to prepare. They are less expensive to produce in the laboratory, and most importantly, in case of RNA vaccines, so there is no risk that the RNA will integrate into the patient's own genetic code. This is what happening with the DNA vaccine. Let us see how the DNA vaccines they are produced. So as I said earlier that DNA vaccine contains the DNA that codes for a specific protein or specific antigen from the pathogen. So when these are injected into the cell, they will direct the synthesis of protein, and these proteins will be recognized as a foreign protein or antigen, and the immune system will get triggered, right? So here you can see how they are prepared or developed. So here a particular gene, it is isolated, it is incorporated into the plasmid of a particular bacteria, right? Then after the process of transformation, these bacteria, they are allowed to grow, and then they are then that plasmid DNA is being isolated, purified, and then it is incorporated into the vaccine. Let us see the advantages of DNA vaccine over the traditional vaccine. So DNA vaccine, so here it uses only the DNA from the infectious organism and not the entire germ or microorganism. It avoids the risk of using actual infectious organism. It provides both humoral as well as cell-mediated type of immunity. And most importantly, it is more stable since refrigeration is not required. As I said, there are a few disadvantages which are associated with DNA vaccine. So it is limited to protein immunogens only. As I said earlier, that there is a potential integration of plasmid into the host genome, leading to the insertional mutagenesis. There can be a chromosomal integration, so chances or possibility of cell transformation. Then there is a possibility of induction of autoimmune response. So sometimes the antibodies, they are developed against the DNA which are incorporated into the vaccine itself, right? So and those antibodies will going to kill the DNA. And the immunogenic response which is exhibited by this DNA vaccine, it is less and it is non-specific. Let us talk about the RNA vaccine. So these RNA vaccine, these are basically produced through the synthetic process by using the certain kind of enzymes. So several advantages, as I said earlier, which are exhibited by RNA vaccine. So they are easy to prepare, they are cost effective, and one more thing is that they are more stable as compared to other kind of vaccine. And so these vaccines, they are also need to be combined with certain adjuvants in order to stimulate or in order to get the enhanced or better immunogenic response. One more advantage of this RNA vaccine is that the very low dose is required in case of RNA vaccine, because most of the vaccines currently which are under development, they are undergoing a self multiplication or self replication. Right, so those RNAs, they are self-replicating, so they are replicating in our body. So the dose for whatever required for generating the immunity, that is very, very low. At the same time, this low dosage equates to mobile manufacturing suits at cost well below the existing vaccine processes. So these are the important steps of the production of mRNA vaccine. So first of all, you need to identify the pathogen, determine the genome. So here the genetic sequence it is determined. Then you need to construct the plasmid DNA. After construction of this plasmid DNA, so there are two techniques you can use. Either you can go with the molecular cloning, or you can go even by in vitro synthesis. And after this, you need to take the pilot vaccine batches. During this process, you need to convert the construct plasmid DNA into the RNA through the process of transcription. And once the transcription is over, once the RNAs are synthesized, you need to do the capping. And after this, you need to subject the synthesized RNA to purification process, and then finally they are incorporated into the formulation. Right, so this is what the schematic representation. So here the target pathogen is identified, genome sequence it is determined. So it is deposited into the database so that it will be readily available throughout the world, and then one can use that genetic sequence for synthesizing the RNA into the laboratory, right? And then this synthesized RNA is incorporated into a formulation, then it is manufactured and then vaccinated. Right. So here the mechanism it is depicted that how RNA vaccine will going to act. So once the host is vaccinated, the RNA, mRNA, it will get entered into the cell, right? So here, when it enters into the cell, it will direct the synthesis of the protein, and these proteins, they are behaving like antigens, and in response to these antigens, the immune system will get triggered, right? So, you will get the protection. So as I said earlier that there is one more type of vaccine. I think one of the well-known company, renowned company, it is undertaking not just one, but there are few other companies are also there which are undertaking the development of this special kind of vaccine, that is what non-viral delivery of self-amplifying mRNA vaccine. Right? So I hope you must be aware about this particular term, self-amplifying, right? So the mRNA which is there in this vaccine, so when it enters into the body, so it will undergo amplification, right? So there is increase in the number of copies of this or mRNA, right? So there is increase in the what you can say number of this mRNA in the human body, and because of this very low dosage of this mRNA vaccine is required. So here you can see RNA is enzymatically synthesized in vitro from the DNA template, then it is injected into the host, and here you can see the RNA self-amplifies in the host, and then expresses the vaccine antigen, right? So this is how it is produced on the commercial scale. So the first important advantage is that it is cell-free synthesis. So you are going to synthesize it from the DNA template to the process of enzymatic in vitro transcription. Right? And then it is subjected to the process of purification, and after purification, you need to sterilize it through the process of filtration, and then you can get that purified mRNA, you can fill it into the vaccine. So obviously you need to add some adjuvants along with this in order to enhance its immune inducing property. Here I would like to put on one example that after determination of sequence of the coronavirus genome, just in 45 days, the first RNA mRNA vaccine it was developed. Right, so very less time it is required, or it is taken up for developing, for synthesizing this particular type of vaccine. Right, so this is how here you can compare the simple synthetic mRNA vaccine and the advanced self-amplifying RNA vaccine. So here from a single RNA, several RNAs have been developed, right? So obviously there is increase in number of antigens, and you'll get a better immune response as compared to the simple synthetic mRNA vaccine, that too in very low dosage, right? Because of its self-amplification property inside the host or human body. So these mRNA type of vaccines, they are not just used for prophylaxis purpose, but they are also having therapeutic applications, especially in the treatment of cancer. For COVID-19, these two examples I have quoted here for DNA vaccine, and two examples from RNA vaccine. One is manufactured by Moderna, and second is manufactured by Pfizer. So both these development those belongs to the category of self-amplifying mRNA vaccine. And under DNA vaccine, so one which is manufactured by Inovio Pharmaceutical, and second is by Genexine. So here you can compare the advantages and disadvantages of viral vector vaccine, DNA vaccine, and RNA vaccine. So as I said, as compared to viral vector vaccine and DNA vaccine, the RNA vaccine, they are very easy to prepare. They are non-infectious. Then they most importantly, they do not integrate with the human genes. Then they don't require the eggs or cells for its preparation. Then they are, as I said, just within 45 days, the first RNA vaccine when developed, right? So it takes very less time. It also having advantage that it stimulates the innate immunity or innate immune response, in addition to the induction of T and B cell immunity. The major concern which is there with this DNA RNA vaccine, even with the viral vector vaccine, is that they are having low immunogenicity. Now let us talk about the recommended timeline, or what you can say the traditional timeline. So if you look at the traditional timeline, so around 15 years, minimum 15 years, 10 to 15 years of time it is required to come to develop the vaccine to the end stage, right? So the first step is exploratory step, wherein the first pilot batch of this vaccine it is being developed. Then it is subjected to preclinical studies. So the developed vaccine it is tested in the animal, right? So here, after the preclinical studies, one has to the sponsor or the developer or the manufacturer has to file the investigational new drug application. After getting the green signal from the regulatory agency, one can proceed for phase 1, phase 2, and phase 3 trials with the developed pilot batches, right? And once the phase 3 trials they are successful, then one has, that sponsor has to submit the entire data, especially the clinical trial data, to the regulatory authorities for getting the approval of the biological license, right? And after So after getting the approval from the regulatory agency, the manufacturer or the sponsor can go for manufacturing of the vaccine. So this entire development, if you will see, so totally it takes around minimum 10 to 15 years. But in emergency situations like a pandemic situation, there some relaxations have been given by some of the regulatory agencies. So you just go through the different phases of the vaccine production. So you can see here for research and development, ideally 2 to 4 years are required, but under the accelerated conditions, the 6 months, and as I said, just 45 days have been taken up for the development of mRNA vaccine. So one can go with the novel or advanced technology for the preparation of vaccine, so that will reduce the development time. The second is pre-clinical preparation. So normally 2 years time is required, but under accelerated condition, it was reduced to 6 months for clinical trials. Ideally, 5 years time it is required to do the complete investigation, but under accelerated condition, the regulatory norms have been relaxed to just 1.5 years. The approval time, the normal approval time, which is taken up by regulatory agency, that was 1 year, but under accelerated condition, it was reduced to 6 months. For manufacturing, the ideal time was, the ideal required time was 2 years, but here it is just 3 to 6 months, and for distribution, it was 3 to 6 months, but under accelerated condition, only 1 month. No doubt here that most of the steps, most of the stages, they have been conducted simultaneously in order to reduce the time duration for studies of vaccines, especially in the pandemic situation like COVID-19. So here the target vaccine production period was around 12 to 18 months. This is how the entire process of vaccine development it is taking place. So initially, one has to do the development in the research and development. So here, the sponsor has to first identify at least 1 to 5 targets. He has to prepare smaller batches. For those smaller batches, one has to do the in vitro testing, especially testing those batches for the immunogenicity. One has to do the target selection during this particular phase, and after selection of the target, one need to validate those selected target by doing certain in vitro testing or in vitro assays, and after successful in vitro trials in terms of in vitro assay or in vitro tests. These prepared small batches can be subjected to animal studies. So obviously you will have to prepare the animal protocol, get it sanctioned from the regulatory agencies, right? And then conduct the trials in the animals. Once the trials are successful, then you can, as I said earlier, that you need to request to the regulatory agency for conducting the clinical trial. Right. So this clinical trial, it is conducted in 3 different phases. In phase 1, the very small number of population it is used to ensure the safety of the developed vaccine. Right. After successful completion of phase 1 trial, you need to go for a phase 2 trial, wherein along with safety, the efficacy of the developed vaccine, it is being assessed, right? So here, especially the side effects and adverse events, the harmful effects or toxic effects, they have been identified. If the vaccine it successfully passes this phase 2 clinical trials, then one can go for phase 3 trials. So in phase 2 trials, the population selected, that is from 100 to 1,000, and for phase 3 trials, this population, it may be from 1,000, from you can say 10,000 to maybe in lakhs. So here you can conduct multi-centered, multi-site trials. So here you're going to study the safety, efficacy, in addition to this, the protection ability which is offered by that particular developed vaccine. Right. So after completion of this phase 3 trial, the regulatory agencies, they will give a license, that is what biological license for manufacturing of the vaccine. Right. So this is how the vaccine it is being developed. Right. So these are the crude steps which are in the production or manufacturing of the conventional type of vaccine. So if you look at the upstream processing, so two important steps are there. First step is selecting the strain for vaccine production, and second is growing the microorganism. And in downstream processing, you can see isolation and purification of microorganism. After purification, inactivation of those organisms, followed by formulation development, then quality control and lot release. This is how the journey of vaccine in the manufacturing facility inside the industry. So here the first step is bacterial or viral. Here the pathogen is identified and it is multiplied or replicated, right? So harvesting is done, purification, then inactivation, then assembling is there, followed by formulation development, filling, that is what packaging and filling, freeze drying is there, followed by again packaging, batch release, transport. So I'll just discuss about the one of the important step which is there in the manufacturing of vaccine, that is what growing of the microorganisms. So for bacteria and viruses, different techniques have been used. Generally, for growing the bacteria, the bioreactors have been used, right? So two different techniques they are used. One is batch culture technique, and second is continuous culture technique. Whereas for growing the viruses, the cell culture or tissue culture technique, or even bird embryo technique, then live animal inoculation technique, and even sometimes transgenic animals they are used. But mostly these two techniques they are used for growing the microorganisms in the vaccine manufacturing. The first one is egg-based technique, and second is cell culture technique. Among these two techniques, cell culture technique is more preferred. Right, so let us see the first technique, that is what egg-based technique. Right, so here I have represented how the process is followed. So here in case of egg-based technique, so you'll have to first isolate, or you'll have to choose a particular virus, and this virus it is injected into the fertilized eggs. Right, so this is what the first step. Second step is incubate the egg and allow for the replication of viruses or the microorganisms. The third step is collection or separation of the allantoid fluid from the egg, right? So this fluid, it contains the live viruses, right? So next step is purification, separation, deactivation, and so on. These are the different sites which are there in the embryonated egg, where one has to inject or inoculate the pathogen or the microorganism. Right. So the first site is this fluoroallantoic membrane. So here basically the viruses, they are injected. Second is amniotic inoculation. So here it is injected. Third site, it is allantoic inoculation, and fourth one, that is what you can inoculate inside the yolk sac. The second technique is cell culture based production. So here I will have to again isolate the pathogen. Right, you'll have to do the cell culturing. So you can go for virus. Here the inoculation followed by harvesting, that is what bulk manufacturing, followed by purification, and then formulation development, so addition of several excipient, then filling, inspection, labeling, and packaging. Right, so here you will going to use the different cell lines for growing the viruses or bacteria, right? And after the culturing, harvesting, you need to subject it for a separation process like centrifugation, right? So here you're going to separate out certain components, and then you can subject it for the preparation of vaccine, right? So these here I have compared the advantages and disadvantages of the egg-based vaccine and cell culture-based vaccine. Right. So here you can see in case of cell culture vaccine, there is a security of supply. Then secondly, the time span which is required, that is just 3 months, whereas in case of egg-based vaccine, so it takes around 6 months. Here the scale-up is rapid in case of cell culture. Best vaccines, here the vaccines can be well characterized. They are having good antigenic stability, and this whole process, it is being carried out under the controlled condition, right? So you can say under the sterile process, right? So that doesn't happen with the egg-based approach. So here there can be a possibility of contamination. Even in certain vaccines, the allergic reactions have been encountered for those which were developed by using the cell-based growth of microorganisms. So these are some of the vaccines which are produced by cell-based technique. Right here, you can see some of the viral vaccines which are developed by cell-based techniques. Some are there which are developed by egg-based techniques. Now, when you look at the conventional manufacturing process, you can see the several steps are there. The preparation of media, right? So this is done openly in the open environment. Right, then cell amplification, this batch culture is there, clarification, then nuclease treatment, DNA removal through the ultrafiltration, inactivation, centrifugation, ion exchange chromatography, then formulation development, sterile filtration, and finally you are getting the drug substance, right? So you just look at several steps. They are being carried out openly in the environment or atmosphere. So more chances of contamination or what you can say, bioburden is there. Right, so this is a major concern, especially with the conventional process of development of vaccine. And the major entry points for contamination is facility, equipment, process, materials, utilities, and personals. You can, if you'll do just Google search, you'll get several warning letters which are issued to several vaccine manufacturing companies just because of inadequate control of bioburden. Here, one warning letter which is issued to GSK that I have represented here. So in 2014, they have got one warning letter because of the inadequate control of the bioburden, and therefore the results for bioburden and endotoxins, they were out of specification. Right. So likewise, several companies have been till date issued the warning letter by the FDA, right? So, so to get rid from these problems, to avoid or to in order to reduce the contamination bioburden, or to get a good quality vaccine. So one approach which is being suggested, that is called as multifaceted approach. So here, so the contamination or bioburden, it is being controlled through three different important steps. One is prevention, second is detection, and third one is removal or clearance. Right. So under prevention, one has to verify each and every raw material which the sponsor will going to use it for the development and processing of the vaccine. Secondly, detection. So whatever entire process which is used for development and manufacturing. So each and every step one need to assess it, test it for the presence or absence of contaminants or microorganisms. So for detection or for testing, one has to have a good or I'll say advanced instruments or equipment or analytical tools, right, for getting the accurate and more precise results, right? And the third approach, which is followed, that is what clearance or removal, right? So so one has to develop the capacity of manufacturing process with orthogonal steps that removes or inactivate the contaminants, right? So here basically you can go with different processing tools like filtration, chemical inactivation, or even you can go with some modern sophisticated chromatographic tools for separation of contaminants from the components of the vaccine. So this is what the entire process you can see in the photograph or picture, which is followed for manufacturing of vaccine. So here at different steps you can incorporate or you can use different steps for clearance in terms of filtration, maybe in terms of chemical inactivation, right? But this will make the entire process time consuming, very cumbersome and tedious. And therefore there is need to change the technology from conventional technique to the modern tool or modern technique, right? So let us see what are the changes which have been done in the conventional technique in order to get a very good quality of vaccine in a very short duration of time. So the first change what is there, that is what single-use technology. This is followed by a closed system wherein no open handling of the product is done. Most of the processes, they are made automated. Even the sampling preparation, it is done with high throughput sample preparation using the compact liquid handling system. As I said earlier, the newer chromatographic techniques have been recommended for purification. Tools like centrifugation have been replaced with the tangential flow filtration for getting better quality intermediate, and high throughput analytical tools like UPLC and several others can be used to get better accurate and precise result. And one can even go for increasing the output of the production by implementing systematic design, like a design of experiment. These are the advanced purification technologies which one can use in order to reduce the production time, as well as to reduce the contamination and the bioburden. So let us look at the benefits of single-use technology and closed system of for flexible manufacturing of the vaccine. So it is time-saving. It increases the process and operator safety. It eliminates the use of flammable or hazardous solvents, which were initially or in the past were used, especially for separation, precipitation, or purification of the different components of the vaccine. At the same time, it is flexible, compact, modular, cost-effective, and disposable. There is improved throughput and reduced turnover time. So it will be reducing the downtime for cleaning, sterilization, and lot many processes which are involved, especially in engineering. So in total, it will reduce not just the cost of the production, but at the same time, it will improve the quality of the output, as well as it will also reduce the cost of production. So this is what representative sterile production process of a live viral vaccine from cell culture, then harvesting, clarification, purification to formulation, right? So entire process starting from the cell culture up to the formulation, it is carried out in a closed system. So there is completely closed system, and that too, it is just for a single use. So once your process is over, you can dispose it off. And so this is one more representative process you can see for this single-use closed system, right? So you can see here the different processes, right? So viral release, then microcarrier separation, clarification, filtration, then ultrafiltration, then DNA digestion, followed by ultrafiltration, and finally formulation development and filling. So everything it is carried out in a closed system, in a disposable system, right? Regarding formulation development, similar to as what we are following for the medicine. So the similar steps have been used here for vaccines. So the development of a formulation that also involves the pre-formulation studies. Right, so here basically the excipients have been selected. So these are the different kind of excipients which are generally added to the vaccine. The first one is adjuvant. So I already discussed about this. So these are added to most of the vaccines as the immune response. Right, so one of the example I have quoted aluminum or alum salt. Right, so in past steps you can get several references where this particular adjuvant was added. Then several preservatives have been added to prevent the microbial growth. Stabilizers are added to protect the vaccine component during freeze drying or during the process of heating. Gelatin, then monosodium glutamate, and several examples are there. One more component is added, that is inactivating agent. So generally formaldehyde, glutaraldehyde, these are added during the process of production. Even antibiotics like neomycin, streptomycin, they are added to avoid the growth of some of the unwanted microorganisms, right? So suspending fluids, they are also added to kill or weaken the microorganisms. So few examples I have quoted here: egg protein and yeast protein. But the examples, they are not restricted to this. You can have several examples, right? And once the pre-formulation studies are over, one has to take the trials, right? One can even go for novel drug delivery systems like liposomes, polymeric nanoparticles, cold nanoparticles, hydrogels are there. Then even you can go for microneedle scaffolds, hydrogels. Right, so depending upon the routes of administration, you can have different novel drug delivery systems, right? So I think some of the drug delivery systems, even they are available in the market, like microneedle, nanoparticles. Right, so currently some of the vaccines which are under development, they are in the form of lipid nanoparticles, when polymeric nanoparticles are there. Right? So this is what the typical composition of vaccine formulation. Here I have just given an example of generalized vaccine formulation. So it contains around 1 to 2% of antigens, around 95% of solvent, and around 3% of other excipients may be required for pH adjustment or for adjusting the tonicity in case of parenterals, sweetening agents for oral formulations, preservatives, antioxidants, then surfactants, even stabilizers and thickeners. Of course, vaccines need to be subjected to stability studies, so you can get guidelines from the WHO in 2006. The World Health Organization come out with certain guidelines related to stability evaluation. Right, the next part is quality control. This is one photograph I have taken from the website of Sanofi. So you just look at the bottom, right? One very important sentence here they have written that 70% of the time of production of a vaccine is dedicated to quality control, which represents several hundreds of tests, right? So not many quality control test or quality control evaluation at different stages of development, production or manufacturing one need to carry it out for getting good quality vaccine, right? So some of the vaccine non-clinical studies have listed out here. So first one is product characterization. Then in case of live vaccine, that attenuation test is there, then inactivation or reversion test, absence of adventitious agent, pyrogenicity, potency, immunogenicity. Then sometimes the challenge test is done, that is what protection studies and GLP toxicity studies, especially for novel formulation. Then you need to subject the vaccine for sterility test, safety, residual toxicity, efficacy, increase in virulence, interference test, consistency in terms of production. So here you need to produce the 3 consecutive batches, and you need to subject the data to the regulatory agency in order to get the approval for the production, then assessing the risk to the environment, and finally the lot release test. These are some of the analytical techniques which can be used to evaluate the vaccine or vaccine intermediate at the different steps of production or development. But this is one representative certificate of analysis. You can see a series of tests are there, starting from appearance, then identification, purity of DNA. So this is especially for the plasmid DNA, right? So you need to determine the purity in terms of protein contamination, in terms of RNA contaminant, pH, then bacterial endotoxin, and so on. Right, so depending upon the different type of drug delivery system formulation, one need to choose the different evaluation parameters. right? And as I said, after filling, packaging, and labeling, right, one has to carry out the field test and performance monitoring, right? So this is how the entire development and manufacturing process of vaccine is being carried out. Now let us discuss about the licensure process. So in order to obtain the permission to conduct the clinical studies, the sponsor has to first prepare the pilot lots for the experimental purpose, right? So this pilot batch, you need to subject it to preclinical studies or preclinical testing in the animals, and then this particular data you need to submit it to regulatory agency through the investigational new drug application, right? So the complete new drug application, investigational new drug application, so it should include the description of composition, source, manufacturing process of the product, quality control, and the methods used to test the vaccines, safety, purity, potency, summary of all laboratory and preclinical animal testing, detailed description of the proposed clinical study, name and qualification of each clinical investigator. Right. So once the regulatory agency is getting satisfied with the data which is submitted by the sponsor, then the regulatory agency will give the preliminary approval to the sponsor for conducting the clinical trials. Right. So, and after successful completion of clinical trial, one can go for a biological license. But here, in case of COVID situation, we have seen that the many regulatory agencies, they have given the emergency use approval well before time. So obviously that emergency use approval, that is conditional approval, and it is restricted to certain limited population. If we look at the regulation pertaining to vaccines in India, so vaccines, they are treated as a new drug, right? So the manufacturer has to obtain the license from the CDSCO for manufacturing, for import and manufacture, for sale. One has to refer Drug and Cosmetic Act and Clinical Trial Rules 2019. The detailed requirements and guidelines for conducting non-clinical and clinical studies and approval of vaccine, one can get it from second schedule of New Drug and Clinical Trial Rules 2019. All the vaccines are required to characterized and manufactured in compliance with the good manufacturing practices as prescribed in the rules. In case of manufacturers for rDNA derived vaccines like mRNA vaccine and even the protein kind of vaccine, so the requirements and guidelines prescribed by Department of Biotechnology one need to comply with. So this is all about the vaccine regulations or the regulatory requirement, right? I know that you all are eager about how soon can we have a COVID vaccine. So during the survey, during searching the literature, I came across several cartoons, right? So you can see two friends, they are sitting 2 meters away from each other and they are waiting for the vaccine. And this is one more vaccine that people, they are not going out. So this is one more cartoon. I'll say this is one WhatsApp message I received on my WhatsApp. Vaccine की राह देखते देखते कोरोना का बर्थडे भी नजदीक आ गया. So everybody is eagerly waiting for the vaccine. This is one more interesting cartoon. You can see here one ICMR scientist is cutting on the vaccine. Simultaneously, here one political leader is delivering his speech, and he's doing some comments, right? So you can see the comment which is done by that political leader. August is the vaccine deadline, right? So you can just look at the expression of this ICMR scientist, right? So obviously it is not a political science, it is a pure science, it is a basic science. So it is not a joke to develop a vaccine in a very short span of time, right? If you look at the previous history, you just look at the development of this polio vaccine, it took around 60 years to develop, right? Even for Ebola virus, it taken around 15 years to develop. The outbreak of SARS coronavirus 1, it was took place in 2003, and around 17 years have been passed away. But till that, there is no vaccine for this SARS coronavirus 1. Similarly, in case of MERS also, the outbreak was found in 2014, and after 6 years, even there is no vaccine, right? And we have tentatively been given just 12 to 8 months to develop the vaccine. So how it is possible, right? So the scientists around the world, they are working day and night on developing the vaccine for the coronavirus, right? So the sequencing, genomic sequencing of this particular virus, it was done well before in December 2019, right? And these three major technologies, they are used by the industries. One is protein-based technique, wherein the spike protein they are purified and injected. The second technique, that is viral vector-based technique, where the spike protein gene, it is purified and it is injected into the adenovirus that is used as a vector, and when it comes in contact with the body produces this spike protein, and you are getting the immune response. And third one is mRNA vaccine. This is a recent development in the this is what you can call novel type of vaccine. So here the mRNA, it is injected, and you are getting so our body produces a spike protein, and then you'll get the response. So these three major category of vaccines, they are under development. This is the report. You can see here that 5 potential candidates till date have been identified, which are manufactured by 5 different companies, which are there mostly in the phase 3 trial, and probably the phase 3 trial will going to complete in 2020. These are the 3 candidates. They have announced the effectiveness of their vaccines. So for Moderna vaccine, they are claiming that their vaccine is effective to an extent of 95%. Then second one is Sputnik V 5, that is claiming around 92% efficacy, whereas the vaccine which is manufactured by Pfizer, that is claiming around 90%, right? So this is the recent data which is collected on 17th of this November. Right? So just by looking at this statistic, one can say that we are very near of the success, and very soon I think probably we can get the vaccine. Right, so I'm ending this slideshow with this that today, if you look at the news which is published in today's Maharashtra time, right? So one of our health minister said that in a span of just 4 months will be with the final vaccine of COVID-19. That will be an approved vaccine, right? So will be available in the market. That is what predicted by the health minister. Let us see how the regulatory agencies, they are responding to this. Until and unless where the sponsors or companies, they are not getting the regulatory acceptance, right? So the vaccine will not reach to the market. Right? So finally, I like to just conclude the session with certain remarks that making vaccines uses complex production methods, meticulous quality control, and reliable distribution channels. Manufacturing technologies strongly influence vaccine cost, ease of industrial scale-up, stability, and ultimately worldwide availability. Large-scale production ensuring proper dosing for efficacy and addressing potential safety concern remain the significant hurdle. Reliable vaccine production in the appropriate quantities at affordable price is a cornerstone in the development process, especially for COVID-19. The safety and efficacy, it is still under question. Let us wait and watch for the approval of regulatory agency. And finally, I'll conclude my session. I'll conclude this today's presentation with one simple sentence that vaccine is not an ultimate solution. So I'll suggest try to boost your own individual immunity rather than relying on vaccine. No doubt, those who are not having good immunity, for those this vaccine is the better option. But those who are younger or even for older also, there are many options available to boost immunity just by following simple natural procedures, right? So do follow those natural procedure and try to boost your immunity so that will not going to require vaccines or medicines to treat such a bacterial or mostly the viral diseases, right? So with this, I'm ending my presentation. So once again, thank you very much to all of you for listening me very patiently. Shruti, I have completed
Yes, sir.
my part.
Yes, sir. That was really a great presentation, sir. And I guess you have covered such a huge topic in a very wholesome manner. Now, looking at the time constraint, with your permission, can we move on to the question and answer session?
Yes, yes, yes.
Okay, sir. So we have a couple of questions.
Okay.
Yeah, the first question is there any limit of conjugating respected number of genetic material to the adjuvants in conjugate vaccine?
Can you repeat the question?
Yes, sir. The question goes like, is there any limit of conjugating respected number of genetic material to the adjuvants in conjugate vaccine?
As such, there is no limit. But while developing the formulation, one has to also see the compatibility of the adjuvants along with the other constraints of the vaccine material.
Yes, definitely. The next question is, can you please explain a bit about herd immunity?
Yeah. In case of herd immunity, so at least 70 to 80% of the total population has to become immune to a particular pathogen, especially in case of epidemic. Then what will happen? The chain of transmission of a virus from one person to the another person, that will get broken, right? And then naturally the virus or that pathogen will die with its own natural death. So that is what happened with the polio virus. We'll look at the past history.
Okay, sir. The next question is, people say that vaccines are linked to long-term health problems such as multiple sclerosis, diabetes, and autism. Is that true?
Yes, in very limited number of cases of population, I'll say these kind of adverse events have been encountered. In my presentation, also I have mentioned earlier that it is quite difficult to just predict what kind of unwanted adverse drug reactions or side effects will be there with the developed vaccine, right? In the upcoming time, right? So being the vaccines, they are not subjected every time for the long-term studies, especially for their safety and toxicity. So these kind of events can occur, right? So this is also happening not just with this is happening not just with vaccine, but even you must have observed with even synthetic drugs also.
Yes. Okay. So moving on, there is another question. How do government regulations impact the process of vaccine development? And another parallel question to that is, does the protocol for these vaccines change if there is an emergency? So as like we have a global pandemic going on. So do the protocols
Yes.
change accordingly? And what impact do the government regulation have on it?
Yes, yes. First of all, I'd like to mention here that regarding the regulations or regarding the regulatory agencies. So even though they have relaxed somehow in the protocol at some point, but they have taken care that there is no compromise in the quality of the final vaccine. Right, so some relaxation in terms of timeline has been given. Especially, they have relaxed the approval, that approval period, what is generally taken in the normal time. Right, they have relaxed to some extent. I think 6 months, 3 to 6 months, they are taking for getting the approval. At the same time, they are allowing some of the studies to do simultaneously, right? Some of the trials to conduct simultaneously. So by doing this, I think the span of development of vaccine has been reduced. But one thing I just wanted to mention here, that regulatory agencies, they are stringent in terms of the quality and safety of the vaccines. So they have not compromised the quality. Otherwise, at this moment, the vaccine was there in the market. So this is the reason
So then,
that there is, yeah.
yeah, go on.
Yeah. This is the reason that till date vaccine is not there in the market.
Yes, sir, but then what are your thoughts about vaccines which are introduced in market on a short notice? For example, Sputnik 5.
I think even though the Sputnik 5 has got the approval, that emergency use approval, right? And but still, I think better candidates they can come in the market, which will be more efficient to this particular vaccine. Some more novel technologies are there. So some vaccines, they are coming out with the new techniques like mRNA self-amplifying mRNA technique is there, right? So some novel adjuvants even have been incorporated at this time during the vaccine development. So we can look forward for getting even some more better quality vaccine. At this moment, whatever vaccines we are getting at the cheapest price, or at what you can say in a small span of time, so we have no options. We have to go with them.
Yes, absolutely. And another question is, how does the vaccine making process copes with the mutations occurring in the virus or bacteria over a period of time till it is introduced in the market?
this is a very critical step. So during, I'll say during each and every processing step, the sponsor or the manufacturer has to take care that the selected pathogen or microorganism that should not come across to such a stress condition where the mutational changes can happen in the microorganism. So there one need to take care. Right? So that is a big topic of discussion again.
Yes, sir. We have another question. At the moment, the world is in financial recession. How would that affect the pricing of the vaccine?
Definitely, everybody is looking forward for a cost-effective and cheaper vaccine. But the cost of the vaccine, it is decided on what technology, what technique they are using for manufacturing. As I said, if you are going with live attenuated vaccines, then obviously the cost will be higher. If you are going with protein-based vaccines or mRNA-based vaccines, the cost will be comparatively lower. I think I came across one of the reference wherein the proposed cost is around 1,000 rupees for 2 doses of one of the vaccine. Right, so obviously government will try, or even the manufacturing companies, they will try to reduce the cost of the vaccine, so that the vaccine will reach to the common community, common people.
Right. Yes. We'll just take a couple of questions before we end the session. What are combination vaccines and why are they used?
Regarding combination vaccine, so there are sometimes 2 or more vaccines, they are combined together, right? And they are given through a single shot. So these type of vaccines, they are called as combination vaccine. I think couple of vaccines, they are available in the market. I think MMR vaccine is there, that is called as, that contains 3 different vaccines. That measles vaccine, then mumps vaccine, and rubella. All these 3 vaccines, they have been combined together. So likewise, you can get several examples. The basic advantage or benefit is this type of vaccines, they are benefited to children, they are benefited to parents, right? So for children, they don't have to again and again take the vaccine. For parents, they don't have to take their child every time, and they don't have to recall or remember every time which vaccine is given, which is not given. Right? So in this respect, it is beneficial to parents. Even it is beneficial to health workers. They don't have to store all these different kind of vaccines. So they can reduce the cost of storage. For nation also, right, when certain immunization programs, they are conducted, so that will obviously reduce the cost which is incurred in conducting such a programs.
Yes, definitely. And last 2 more questions. Can the sale of orphan drugs be possible at generic drugs price while overviewing COVID like a pandemic?
Regarding orphan drugs, I think regulatory authorities have given a lot of relaxation, right? So maybe in terms of the cost of approval, or maybe in terms of the process of approval, right? So I think that one can get that particular benefit. So especially for the orphan drugs. And even the governmental agencies, they can give some relaxation, particularly to orphan drugs.
Okay, sir. And the last question, and I guess most of us really want to know the answer for this question, is that how long will the vaccine, let's say COVID-19 vaccine, protect patients, and will it protect the most vulnerable patients as well?
So again, I'll say that what kind of vaccine it is and what type of vaccine, whether it is live attenuated vaccine, whether it is subunit vaccine, or it is sterile type of vaccine. Right. So based upon that, the time period will depend. Live attenuated vaccine, it gives what you can say. It offers immunity protection for longer period of time, right? So generally, the licensing authorities of the regulatory agencies, they give license, or they approve the license only if the product or the vaccine it is giving or it is giving protection at least for 1 to 2 years, right? But now at this moment, it is quite difficult to wait for 1 to 2 years to
Right.
wait for all this complete process, right? So again, that varies with the vaccine to vaccine, right? So that will be from 3 months to, I'll say, can be up to 1 year, 2 year, or 5 years, or maybe a lifetime, right? So that so in upcoming time, after the post-marketing surveillance, will come to know the real picture of these vaccines, right? So even
Surely.
some more companies are there, even though they are claiming, I don't think what will be the future of those companies.
Right. So these were all the questions we had. And now, as we are about to conclude our session, I request Student Exchange Officer Mr. Gaurav Zope to speak a few words. Uh, Gaurav, you are not audible. Could you please check your settings?
Hello, am I
Yeah.
audible right now?
Yes.
Yes.
Now, please continue. We started.
Yeah, so I'd like to start it from the scratch. Okay, so thank you, Shruti. On the behalf of IPA Students Forum, it's my privilege to propose a vote of thanks on this occasion. My heart fills with a lot of gratitude and respect for our distinguished guest speaker, Dr. Harshal Pawar, for not only sparing his invaluable time for us to grace this session, but also for enlightening us with his commendable talk on the subject. Thank you, sir, for clearing the concept and enhancing our understanding pertaining to the vaccine development process. You have indeed put best of your efforts to make this session a remarkable one. Really, sir, we are highly obliged. Next, I would like to thank Dr. Anand Shedge, our respected mentor, for leading us and inspiring us in working towards the goal of conducting National Pharmacy Week program. We are thankful to him for his huge support at all times. We are grateful to Swaroop Agrochemical Industries for being our presenting sponsor and supporting us for the whole pharmacy communal sessions. Conducting the pharmacy communal sessions wouldn't have been impossible with the zealous efforts of Student Exchange Office and Sports Committee, who has devoted a lots of efforts and energy for this program. Furthermore, let us have a special recognition for all of our hosts, that is, Ms. Shruti Kulkarni, Mr. Aditya Davre, Ms. Sahana Mankame, and all our volunteers, Ms. Yukta Deswalkar, Mr. Rohan Chaudhary, Mr. Praveen Tak, for providing their help at registrations as well as for the technical support. And yes, also our PR member, Ms. Vanshnavi Prajapati, for covering all this event on the Instagram page. This vote of thanks would be largely incomplete without expressing my gratitude towards other IPMSBSF members who have been working really hard and carrying out all the allotted duties very diligently. Last but not the least, thank you for being a wonderful and patient audience. We hope to see you tomorrow at the same venue, at the same time, but with a different topic and a different link for joining the session. Thank you again, all of you. Thank you, sir.
Thank you very much to all of you for bearing me.
Thank you so much, sir. So we declared this session to be over.
Thank you, sir.
Thank you so much for joining. Yeah.